Welcome back. We now turn to the second set of presentations. To remind you, we will have a group discussion and audience Q&A following these two presentations, and you're encouraged to submit questions at any time using the Slido link provided on the screen behind me and posted in the chat of the Zoom webinar. Our first speaker of this session is Dr. Étienne Holder, postdoctoral associate at the Boston University Chobanian and Avedisian School of Medicine and Slone Epidemiology Center. Dr. Holder's talk title is Place and Health, Exploring Neighborhood-Level Influences on Breast Cancer Survival. And our final speaker of the program will be Dr. Laramie Duncan, assistant professor at the Department of Psychiatry and Behavioral Sciences at Stanford University. Dr. Duncan's talk title is Fundamental Insights about the Biology of Psychiatric Disorders. It's now my pleasure to invite Dr. Holder to the podium.
Hello and good afternoon, everyone. My name is Dr. Étienne Holder, and I am a cancer epidemiologist by training. And today I'm going to talk to you all about place and health and my work to enhance the health of Black women. And specifically, I'll be talking about neighborhood-level influences on breast cancer survival. So as an agenda for my talk today, I'd like to start by naming some of the US-based women-centered cohort studies, provide some current statistics around breast cancer, discuss the structural drivers of health, talk about my work with neighborhood and breast cancer survival, and talk about some next steps for public health. So there are several long-standing seminal cohorts that have empowered women and enhanced women's health over time. One of the first is the Nurses' Health Study, which began in 1976. I had the privilege of working with the Nurses' Health Study for my dissertation research. And that one's housed locally right here at Harvard University. The Women's Health Initiative, the Black Women's Health Study, which is housed at Boston University, the Sisters Study, and the California Teachers Study. Now, these studies have really laid the groundwork for some newer generation studies, such as Aspire, which is a first-of-its-kind study aimed at recruiting individuals from Asian backgrounds, as well as Voices of Black Women, which is aimed to recruit 100,000 Black women across the US. I have the privilege of being a postdoctoral associate with the Black Women's Health Study, and so I'd like to take a moment to tell you a little bit more about that cohort. The Black Women's Health Study was founded in 1995 among 59,000 self-identified Black women. They were recruited through subscribers of Essence magazine as well as professional organizations, and in 1995, the women's ages ranged from 21 to 69 years, with the median age being 38. They were also recruited from 17 states at the time, but now our participants live in about just over 40 different states across the US. And we get information from our participants through follow-up questionnaires every 2 years. Here's an example of one of our questionnaires that we'll send out to participants. They can be completed via paper or online. We're able to gather information about demographic factors, anthropometrics like height and weight, reproductive history, also the personal medical history as well as their family history. We can also garner information on lifestyle factors such as smoking, physical activity, as well as psychosocial factors such as the individual's experiences with racism and depressive symptoms. We can also get information about medication use, among many other things. The Black Women's Health Study is a wonderful data repository, and we're able to link with several other different organizations, such as cancer registries. We currently link with 29 cancer registries, 29 different state registries that encompasses more than 95% of where our participants currently reside. We're able to link with Medicare data as well as US Census data, so we can learn more about the neighborhood environment where the participants live. And we're also able to link with the US Environmental Protection Agency, so we can learn more about the environmental exposures our participants are encountering. We also have a biorepository where we have donated saliva samples and blood samples. Allows us to investigate more about the RNA or DNA, as well as some important biomarkers like vitamin D levels. We have tumor tissue blocks, so for example, we can investigate more about the tumor microenvironment of a breast cancer tumor per se, as well as a digital mammography repository. So we're able to study those important, well-established risk factors like mammographic density in relation to breast cancer risk. So let's talk a little bit about some of the current breast cancer statistics. So breast cancer is the #1 most diagnosed cancer among females worldwide. And this slide here shows the World Health Organization world map depicting the new diagnoses of cancers across the globe. The countries that are depicted here in darker blue indicate a higher incidence. More diagnoses of breast cancer are occurring. So for example, there's Australia, there's some countries in Europe, as well as Argentina and South America, and North America. And if we zoom into North America, focusing on the United States, we can look at this CDC map that shows the distribution of new cases across the US. Now in the US, for every 100,000 women, 134 are diagnosed with breast cancer. And if you pay attention to the map, the areas that are in darker blue indicate states that have higher incidence of breast cancers. So that's in areas in New England, we have some in the Midwest, as well as Washington State. And pay close attention to where those darker blue states are, because we see a shift when we look at the distribution of mortality. The darker states end up shifting southward, whereas southeastern region, as well as some of the western states, are more readily impacted by higher mortality rates. Now, breast cancer is a very survivable disease with early detection and breast cancer treatments, but it's still the second most, women are still, who are diagnosed with breast cancer, it's the second most cause of female-related cancer death in the US. However, for Black women, it's the #1 cause of female-related cancer death. This bar chart here shows breast cancer incidence, so those new cases, new diagnoses of breast cancer, as well as death rates by race. We have non-Hispanic Black women represented, as well as non-Hispanic white women. And we can see that Black women are diagnosed with breast cancer slightly less often than white women are. However, they're more likely to die from their breast cancer. So the difference between these two bar charts is a 40% difference in mortality. So Black women are 40% more likely to die from their breast cancer than their white counterparts. And this is an area of interest for myself, as I'm interested in teasing apart this relationship and really getting down to those root causes that's driving these types of statistics. One of those areas that I'm interested in exploring further is the social structural drivers of health. So to define that, the World Health Organization defines the social and structural drivers of health as the non-medical factors that influence our outcomes. So those are the conditions where we're born, where we grow, where we work, live, play, and pray. The built environment feeds directly into that, and the CDC defines that as the human-made design of the physical environment. So that's the buildings, like the one we're in today, the streets, open spaces, as well as other infrastructure. So this is a nice schematic that shows a well-thought-out public health vision for the built environment. You can see that there's green spaces, there's a walking trail, a bike path, there's public transportation, and it looks like there's even housing close to markets and things of that nature. And while this is a nice vision for the future and how we should move forward in planning our communities and our environments, we have to understand that it hasn't always been this way, that a lot of the built environment and the structural drivers of health that created our communities were rooted in systematic racism and structural racism. And an example of that is redlining. So redlining started in the 1930s with a federal organization named the Homeowners Loan Corporation. And this organization was charged with creating federal residential security maps for the majority of American US cities. And it's important to note that by definition and by implementation, redlining is the intentional discriminatory practice of community disinvestment. So let's zoom into this figure here. So this is an image of the Hulk security map of the Boston area, adjacent to where we are now. And just to go over the color schema, grade A represents best, and where communities were readily invested in. Grade B represents desirable. C indicates a declining area. And the red areas were literally redlined with a marker and given a grade D, considered hazardous. And it's important to note that in grading these different areas and in deciding where investments would happen, a big portion of what was determined redlined and why it was determined hazardous was because of the racial ethnic makeup of that residence. So communities that had a higher proportion of Black residents were considered hazardous and were intentionally disinvested in. Now, redlining was in the 1930s. That's some over 80 years ago about. But there are still historical impacts because of how this federal organization designed communities and intentionally disinvested in these communities. Some of the impacts of historical redlining that we see 80 years later include the following. So 3 out of 4 neighborhoods that were redlined as hazardous are currently experiencing economic disadvantage. There's still persistent residential segregation in many of these areas, and gentrification is leading to greater economic inequality. It's leading to pushing out of the people who live in those communities, and in an event, just leading to additional persistent residential segregation. Notably, some of the southern states that were designated as hazardous by these redlining maps show the least change today in those communities. And that brings me to my work and the most recent work on neighborhood concentrated disadvantage, individual experiences of racism, and breast cancer survival, where I try to really take into account the economics and social environments where the women live and see how that impacts their breast cancer survival. So this study was conducted in the Black Women's Health Study among 2,290 Black women with breast cancer. So for the Black Women's Health Study, breast cancer survival is determined with linkages with the National Death Index, where we're able to garner information on cause of death. And once women were diagnosed with breast cancer and enrolled in our study, we followed them forward until 2022. We had several different measures of area level indices. This one here is neighborhood concentrated disadvantage, which takes into account the economics of the neighborhood, as well as some of the social implications for that neighborhood. We also looked at neighborhood socioeconomic status, which is more focused on the economy of that community and different factors that impact the economics of the neighborhood. Both of these measures were measured at the block group level, so we were able to see a really honed-in view of what the neighborhood environment looks like for where the women in our study reside. We additionally looked at index of the concentration at the extremes, also referred to as ICE, and ICE takes into account the number of privileged individuals in that community, the number of deprived individuals in that community, all based on economics. And it ranges from 1 to -1, 1 being extreme economic privilege and -1 being extreme economic disadvantage. There are other versions of ICE which are worth mentioning. You can take into account race, but for this project, we wanted to focus on the economics of that neighborhood and that area. ICE is measured and determined at the census tract level, so we had this measure to now give us just a little bit more of a bird's eye view on the surrounding area where our participants lived. Both of the neighborhood level measures as well as ICE were set at the time of breast cancer diagnosis. So let's take a look at some of our findings. Here, this is a survival curve. And so survival curves show us the probability of an event from an event, such as breast cancer mortality. Everyone starts at 1 at the time that they're diagnosed, and each line represents a different quartile or category of neighborhood disadvantage. Things that we look for as epidemiologists and others who analyze survival curves are divergence of the lines from 1, being full survival, and divergence from the lines from each other. Now in this graph, the blue line represents women who live in areas of least disadvantage, meaning that they live in more privileged areas. The brown line represents women who live in most disadvantaged areas. And what we see is that we see a larger divergence over time, over the course of our follow-up time, from these two groups, indicating that women who live in most disadvantaged areas are more likely to die from their breast cancer than women who live in areas of more privilege. And it's important to note that even after we were able to analytically account for important factors towards prognosis, such as breast cancer stage at diagnosis, individual education, and cancer treatments, Black breast cancer survivors who lived in areas of disadvantage experienced nearly a 50% higher mortality compared to Black women who lived in areas of privilege. Our next results look at ICE, and we see something similar. So our survival curve is set up the same. We have the blue line representing extreme economic deprivation, and the green line representing extreme economic privilege. And we see over time that wider divergence from each other and from one. And this shows on this survival curve that Black breast cancer survivors who live in extreme economic deprivation experienced higher mortality compared to the Black women living in extreme privilege. Now, another pillar of my work and my areas of interest are psychosocial stressors, particularly experiences with racism. And the Black Women's Health Study is uniquely positioned to garner information and ask these questions about experiences of racism and discrimination. And so on our questionnaires, we've asked participants to recall their experiences with racism at 3 different time points. We asked them about their day-to-day experiences, so those interpersonal interactions, as well as their experiences with racism in institutional spheres, such as on the job, getting housing, whether it be renting, buying, or mortgages, as well as with the police. And when we analyzed experiences with racism with breast cancer survival, we didn't find evidence of an association. However, there may be several reasons why that was the case. For example, after a woman is diagnosed with breast cancer, there may be other important prognostic factors more specific to the neighborhood environment, the environment in which they're living, that could drive breast cancer survival. But importantly, the Black Women's Health Study has found associations with experiences of racism and other health outcomes, so I do believe that more work is needed to further tease apart and interrogate this relationship. So to summarize the main findings, women who lived in extreme economic disadvantage and concentrated disadvantage experienced a nearly 50% increased risk of mortality from their breast cancer as compared to women who lived in more areas that were privileged. So there's definitely attributes of the environment that we can further tease apart and interrogate to figure out what's really driving this to help improve public health intervention. And I like to think about different public health interventions when I think about the work that I'm doing, because I want the work that I'm doing, although it's population-based, to really impact the individuals that I'm intending to serve. And so when I think about where we can go from here, we just looked at some slides and talked briefly about how intentional community disinvestment has really impacted health and impacted communities some 80 years later. So I think one of the important first steps is intentional community reinvestment in those areas. So that 80 years from 2025, we're looking at some of the positive impacts of having a well thought out, a public health focused neighborhood and community. I think it's also important to research and leverage the positives and the beneficial components of living in these neighborhoods as well. For example, ethnic enclaves. We can leverage what we're learning from these neighborhoods while we're trying to reinvest in them, so that the public health messaging can also come from a positive lens. And with that, I would like to again thank the organizers. I would also like to thank our participants of the Black Women's Health Study who have been dedicated and are very engaged over 30 years. I'd also like to thank the Black Women's Health Study research staff and PIs. I'd like to thank our funders, the Susan G. Komen Foundation, Aspire Grant, and National Cancer Institute, which is funding my postdoc and my work. And I'd of course like to thank my mentoring team, Drs. Kimberly Bertrand and Dr. Julie Palmer, for their guidance and support. And I'd be remiss if I didn't thank Ms. Elizabeth Rhodes. She is our patient advocate on this work, working with us and the Susan G. Komen Foundation to really ground the research that we're doing with the patient perspective, and that's invaluable. And I'd like to thank you all in the room and online for your attention, and I look forward to an engaging discussion.
Thank you, Étienne. So yes, I have to give lots of thanks to the Black Women's Health Study because I am continuing to do work with the Black Women's Health Study and can't underscore the value of these cohorts that Étienne mentioned, just the local ones are the Nurses Health Study, the BU, but the California Teacher Study. no one would deny that the collective work of these cohorts have enriched and informed so much of the risk factor information, not just for breast cancer, but for chronic disease writ large and lifestyle. So what I notice very blatantly from your disadvantage curves is something that I'm sure when we do a fuller discussion, we can bring up with Tara, with uterine cancer, which is what I study, and I'm sure ovarian cancer. But is it a simple thing to say that it's economics, that the curves start the same and then there's this divergence? I mean, what's your opinion on that?
Yeah, that's a great question and great observation. I think that, the first 5 or 7 years or so of those survival curves and after breast cancer diagnosis is really driven by biology, being able to detect the cancer early, get the right treatment, finish the course of the treatment. And then over time, we want to be able to extend life after a breast cancer diagnosis or any cancer diagnosis, terminal diagnosis, as long as possible. So we see that there's other factors that come into play. And in this case, there are some other neighborhood factors that may include individuals' ability to meaningfully engage with their environments. We know that physical activity, for example, is important to survival after breast cancer or any cancer. It's important to the prognosis. So if you're unable to meaningfully engage in physical activity in your environment, perhaps it's not safe, then that can potentially impact those curves and why we see that wider divergence. Because we can imagine that areas and neighborhoods that are more privileged have more resources to have, like nicely paved sidewalks and sidewalks in the first place, and maybe safer streets for individuals to engage in physical activity.
Yeah, I'm reminded of, I'm sure you're aware of Michelle Holmes' study on breast cancer mortality and physical activity, as well as other researchers just looking at the built environment. And it seems a relatively easy ask to make an environment safe enough so people can get out and walk, and in particular, so what do you think in your experience, having done the work with all these great mentors, where's the real resistance?
That is a great question, and I mean, it seems like it would be a relatively easy ask, especially if you're going to have a federal agency intentionally disinvest in communities. Why not, we see the health impacts, so let's now reinvest in those communities, kind of hit the ground running, but I think, I'm not quite sure where the stalemate kind of comes into play. I mean, I think that's, one, it has to be a priority.
Right.
And, times are tough right now, I don't know. But yeah, so I think as long as it's a priority for individuals, and there's a lot of community level grassroots organizations that are doing the work, and so I want to underscore those as well. So I think it needs to be, top down as well as bottom up approaches can really improve those aspects and just have safer areas for people to engage with their environment. Yes,
and a lot of these areas are also food deserts. I mean, it's all correlated around this. It's not dissimilar some areas are worse than others, like in the South, but all roads lead to these kind of environmental ways in which we can help address some of the disparities with respect to survivorship with breast, ovarian, uterine.
Mm-hmm. Right, and while the public health interventions may seem large, I think we can just kind of chip away slowly at the block, make small changes. So you mentioned food deserts. Let's try to incorporate areas or institutions or grocery stores that offer fresher fruits and vegetables, more options for individuals to engage, with healthy eating and healthy lifestyle. So just a small chipping away at the ice block, I think, is what's needed.
Well, thank you very much. I don't want to take time away from our next speaker, but we will talk more, as they say, during the Q&A. Thank you. It's my pleasure now to invite Laramie to the podium.
So hello, everyone. Thank you so much, especially to the organizers for this really outstanding event. I'm so happy to be here, and I'll start off by saying that I'm going to shift gears just a little bit now and talk about mental health and talk about it very broadly. A lot of my work is focused on different populations, but now I'm just talking about mental health very broadly. I'm so excited that now is the time that I'm giving this talk because we really have had huge discoveries and breakthroughs in the field of mental health just in the last few years. I think by the end of this talk, you'll be able to see why these three key sets of discoveries have really put us in a new place in terms of how well we can understand psychiatric disorders. Okay. Great. So I don't think that I probably need to explain to this audience the importance of mental health research. Really, all of us have probably been touched in one way or another by the experience of a mental health disorder among a friend or a relative, or personally. These disorders are incredibly prevalent, and there's good information available about these disorders. I'm going to focus on schizophrenia. This is a condition that is very important to me. It just really pulls at my heartstrings. This is a nice article talking about schizophrenia and how it impacts people around the individual that has schizophrenia. So this is a condition that is characterized by people experiencing things that other people don't experience. People may hear voices, they may have beliefs that other people don't hold. Imagine for a minute if you believed that people were trying to poison you, or if you were hearing voices that were telling you terrible things about you, or that people were out to get you. Imagine how difficult that would make life. Those are just some of the symptoms of schizophrenia, the so-called positive symptoms, but those aren't even the most impairing symptoms. It's actually the cognitive and the so-called negative symptoms of schizophrenia that cause the most problems. Now, for these conditions, for all mental health conditions, all of the medical treatments, the medicines that we have to date, were discovered by chance. It's really remarkable. I think it's incredible that they really work at all, but it's true. They were all discovered by chance. So I think we can agree that these disorders are very serious. They're inadequately treated. One of the big problems to developing new treatments is that we don't know the underlying mechanisms. So just to explain and really to hammer home this point, imagine trying to fix a clock if you didn't know what was inside. If you didn't know if it was gears or electronics or a biological system. In many ways, this is really the situation that we've had for psychiatric disorders. Now, I'm talking about a microscopic scale, about the cells and what's happening within those cells in the brain. That's the level of resolution that we need to understand what's happening with psychiatric disorders. And these mechanisms have not been understood. They are not understood. And so if you're trying to fix a clock and you don't know what's inside, I really think it's remarkable that our medicines work at all. The good news going forward, since we have this complexity of the brain that isn't currently understood, at least in terms of the cellular parts and how they work together, is that there are these recent discoveries that have really dramatically advanced our understanding of things. So I'll start by talking about the fact that we now know many of the genes that influence psychiatric disorders. I'm using this term here loosely. I'm just saying genes. If this were a geneticist audience, I'd say things quite slightly differently, but the sense is absolutely correct. We do know what the genes are for psychiatric disorders to a large degree, and I'll show you how that information was obtained. We also know the cells, and the work that we've done in my lab recently is to put these two resources together in a way that has given us a really dramatically different view of psychiatric disorders. So I'll start by talking about how we know the genes that influence psychiatric disorders. You may have heard of this term, a genome-wide association study, GWAS. It's a very simple study design. So this is a study design in which you aggregate a collection of people that has your disorder of interest. So if it's depression or schizophrenia, you identify people that have that condition, and then you identify a group of people that are the matched controls that don't have the condition. And you compare those individuals in terms of their DNA and specifically places along the genome where it's known that people have small differences from one person to another. These days, when we conduct these studies, we typically look at about 10 million locations across the 23 human chromosomes. So when I was in Boston doing my postdoc, this is the type of work that I did. I worked within the Psychiatric Genomics Consortium and did these genome-wide association studies where we use an analytical framework to examine about 10 million locations across the human chromosomes. So when we finish these studies and we have our set of results, one of the first things that we need to do is to take a look at the results. And we do that with these Manhattan plots, as they're called. These are just scatter plots. There's about 10 million points in this plot if we show the whole plot. And it's a really simple idea. If you look along the x-axis along the bottom, those are just the human chromosomes. And the spot along each one of those alternating gray and black locations is a different spot on a different chromosome. And so we're just showing now where these genetic variations are in the human genome. And then the height of those points represents the statistical significance of that variant, meaning does this variant influence a person's, or is it correlated with a person's likelihood of having schizophrenia? And one of the things that's really important about GWAS is that this is a community of researchers that has been incredibly serious about tackling the false positive problem. In much of medical research, I think you all know that there are a lot of false positives, a lot of results that we can't trust that are just inadvertent findings that won't stand the test of time. The GWAS community was incredibly serious about this problem, and in fact, it's part of the reason that I went into this field of research. The threshold for statistical significance is incredibly stringent. This work was conducted through a worldwide effort. And really, it was Patrick Sullivan at the University of North Carolina that started the Psychiatric Genomics Consortium. Kirsten Conan and others, so many others, are leaders within this consortium that ultimately brought together thousands of researchers from around the globe and millions of participants in these studies to finally discover which genes are linked to these disorders. So I'll show you how that came about. And just to impress upon you what a huge effort this was, if you look at the results from these studies from 2009, this study of schizophrenia didn't identify any genes linked to schizophrenia, even though the sample size was 7,000. So that, for many fields of research, that's a lot of people. But in GWAS of schizophrenia and depression and other psychiatric conditions, that is not enough people. And that's part of what was so important about the Psychiatric Genomics Consortium, is that they realized that we needed to continue to increase these sample sizes. Up until the most recent sample size for schizophrenia GWAS is over 300,000. For the depression GWAS and anxiety disorder GWAS, it's over 1 million people that have taken part in these studies. And so this is just that's what it took. We don't define the effect sizes for something that we're studying. Mother Nature does. So we just paid attention to that and eventually amassed the sample sizes that were necessary to see which genes were linked to these disorders. So I'll just pause by saying this is really good. This is really a triumph in science and in mental health research that we finally know the parts of the genome that are linked to these conditions. And that's so important because the genome is the catalog of biology, so we're knowing the parts that contribute to these disorders. It turns out that that wasn't enough. We hoped that this would be enough. It was not. It actually just raised a new problem, especially for psychiatry. It turned out that interpreting these loci was nearly impossible from the beginning. These did not point to the obvious genes that we expected to find. They pointed to totally understudied or unstudied genes. So we have the genes, that's great, but it turns out that we actually need more to interpret the GWAS results for psychiatric disorders. And that's where this next type of study that comes in. This is really just beautiful science. This is not my own work. This is just work from different researchers around the globe that have used this technique, this single nucleus RNA sequencing. And I'll just give you the highlights of what this work does because it's just really exciting. So if you think about your body and you think about your heart or your fingers, those cells are incredibly different from one another, and yet they have the same genome. So why are the cells different? Well, it's different because different sets of genes are being used in different amounts in those different cells. It's really, that's the basic idea. That's gene expression, and we can measure that in tissues throughout the body. And we can also measure that in the human brain. So in this study, I apologize that this is fuzzy, but I think we can maybe see it more clearly here. In this image, this is a study of the human brain. It's the most comprehensive one to date, where they sampled 105 regions from across the human brain to be able to identify the different cell types that existed. If you talk to neuroscience researchers, world-leading neuroscience researchers, even just a couple of years ago, many of them would say that there were probably a couple of dozen cell types in the human brain. Many researchers still say that. It turns out that there are many, many more. There are at least hundreds. There are probably a few thousand different cell types in the human brain. And these are the studies that showed us that fact. I wish I had more time to explain more about it, but the basic idea is that when you measure the gene expression from individual cells, and then you computationally analyze those data, all of the known cell types are just clearly apparent in the data. They just fall out of the data. But then lots of more nuanced, more granular cell types are also evident. And those cell types are consistent across experiments and oftentimes even across species. So it's showing that this approach is giving us the parts. It's giving us the cell types in different organs throughout the whole body and throughout the whole brain. I think, these types of studies are being published in the top scientific journals every week these days because this technique is just clearly so effective. I think it's especially important, perhaps most important for human brain, because we just didn't know what the parts were until just a couple of years ago. So knowing these 461 cell types, this is not a complete list, but it's a good start. And it's a good representative sample of the cell types in the human brain. So that's what I'm going to focus on now. I constructed this colored bar here. That actually is made up of 461 different vertical lines, each representing one cell type. I made the gray lines represent non-neuronal cell types, and the colored lines are representing neuronal cell types in the brain. There are 378 different neuronal cell types here. And then there's other cells like immune cells and vascular cells that are in the gray portion of the plot. So I'm going to keep showing this bar so you can just know that on the left there are non-neuronal cell types and on the right there are neuronal cell types. So now we have these two incredible resources, and it's just good timing that these two resources became available around the same time. What we did is we put them together to try to interpret the psychiatric GWAS results by using the cell types, the cell type atlas, and asking essentially which of these cells, of these 461 cell types, which ones preferentially use the genes that are associated with schizophrenia, and which ones preferentially use the genes that are associated with depression and Alzheimer's disease and other conditions. And will that lead us to the cell types that are truly associated with these disorders? So we constructed an algorithm. This is a very savvy audience. You'll know that, of course, you can construct an algorithm. The important thing is, does it really work? So that was the big question. We've done years of work trying to see whether or not this approach is sensible. And I think that it is. As far as we can tell, and people reviewing our work can tell, it seems like it is. So I'll show you how the results look now for psychiatric disorders. I'll orient you to this plot that I'll be showing. I'll make it different sizes on different slides, but this idea is always the same. I'm showing, again, that bar of different colors. Those are these 461 cell types, and higher points are more significant, and anything above that line is statistically significant, meaning that that's a cell type that's associated with the condition that we're studying. So first, we wanted to know whether or not we could get true positives when there were indeed true positives from the scientific community. So for Alzheimer's disease, one of the types of cells that is widely thought to be associated with Alzheimer's disease is microglia, and that is indeed our top cell type. Somatostatin interneurons are also significant, and that's one of the top results. For multiple sclerosis, this is an autoimmune condition caused by aberrant T cells and B cells, and those are our top 2 results for multiple sclerosis. So you can see here that when conditions have non-neuronal cell types that are causally associated with those conditions, we do identify them with this set of cells and our algorithm for combining it with the GWAS results. So those are good, true positives to see that the method seems to be working. When we turn to 2 other phenotypes, sleep duration and alcohol consumption, I turned here to neuroscientist colleagues to find out what neuronal cell types were linked to these phenotypes in animal studies where you can causally manipulate things. And these cell types, especially for alcoholic drinks per week, there are D2 medium spiny neurons that you can manipulate to turn alcohol consumption up and down in rodents. So that's a very strong true positive, same with the sleep duration neurons. So basically what we're seeing is that we can identify both non-neuronal and neuronal cell types that are linked to these disorders. And so it seems to be working. And for the sake of time, I can't show the other work, but we've tested this in lots of other ways as well. So now we have this systematic approach. We have really well-powered data sets. That's really key too. We also showed that you need adequately powered data for this approach to work. So now let's look at the schizophrenia results. And this is what I was so excited about. This is what we really wanted to do. We wanted to see what the results would look like for schizophrenia. And this is what we find. It turns out that we find 109 different cell types that are associated with schizophrenia. They're all neuronal. In terms of, at least our first pass results, this is what we see. There's so much that I could tell you about these cell types, and there's so much that we still don't know about them. These are brand new cell types that weren't even known 2 years ago, so they're very much unknown. But I can give you a few high-level findings about these cell types. So one way that we can describe them, one thing that we can say about them is we can ask where are they located in the brain. These are the top 3 regions of the brain where these cell types are located. So they're located in the cortex, the outer part of the brain, the amygdala, a fear center of the brain, and the hippocampus, a structure that's especially important for memory. Those are the top 3 brain regions that harbor these cell types. Those happen to also be 3 brain regions that are known to be implicated in schizophrenia, if you look at imaging studies and other types of studies. So I'll turn now to sort of future directions and what we're so excited about. We can look at these schizophrenia results and ask which cell types are modulated by currently available schizophrenia medications. Even though the first, second, and third generation antipsychotics were discovered by chance, researchers were able to figure out how they believe that the drugs are working. It's through the modulation of D2 dopamine receptors and the striatum. That's the caudate and putamen. That's probably this cell type right here, number 222. And so you can see that is one of the significant results here. You'll notice very clearly also that it's not the top result. It's not one of the strongest results. So now what we're so excited about is the fact that we can think about new treatments that might modulate some of these other cell types that seem to be linked to schizophrenia. Some of these are probably already modulated by currently available therapeutics, and others are probably not. So that's part of what we're doing now. We're very excited to think about new opportunities for treatment development. So just to recap, the reason why we're so excited and we're rushing into the lab these days is that we finally have a roadmap, essentially. We finally have a set of neurons that we believe are linked to schizophrenia. And we have the similar results for depression and bipolar disorder and PTSD and lots of other conditions. So we want to learn all that we can about these neurons and figure out how they function together, figure out what it's telling us about the mechanisms underlying these disorders and underlying what happens when brains are working properly. So that's very exciting. In terms of treatment relevance, as I said before, this is the old treatment target, the main treatment target for schizophrenia medications, and we're excited about the new opportunities now that we have clearer ideas of what to target. So in closing, the basic idea is that we didn't know the parts that were relevant for psychiatric disorders just a few years ago, for schizophrenia, for depression, for other disorders. And now we have a good sense of what the parts are, which ones are most important, and we need to figure out how they work together. So I'd like to say that psychiatric disorders are still serious. They're a bit less mysterious. This is why we're so excited about new treatments. And this work is thanks to so many collaborators and fantastic people in my lab. And thank you all for your time.
Thank you, Laramie. That was great. Lots of work. I had a very naive question, if I may. So I would assume that given I've done a lot of GWASs myself and sort of transcriptomics, I would assume that those results that you showed would apply to both men and women.
It's such a great question. Yes, thank you. So that's such a, is this one? This is such a great question about whether these apply to men or women. I have to say that one of the most surprising things from GWAS studies of psychiatric disorders is that we find that the genetic effects for men and women are almost identical. That was not what we expected, but that's turned out to be true. These cell types, we still need to look and see which ones might be a little bit different in men and women. That's something that we don't know yet, and we're excited to look into.
Right, so the implication would be that it's either epigenetic, or not DNA genetic, but above the epigenetic, or something else, right? I like to say. So is there any idea? I mean, because there are differences, right, with psychiatric disorders, that you see the difference between women and males and females.
Such an excellent point, and I think one of the biggest mysteries, certain disorders are more common in men and others are more common in women. If the genetics are the same, that's not the main explanation. So yes, what is it? What societal factors are relevant? What hormonal factors are relevant? So we're just getting to the beginning of those questions. Yeah.
I have another question related to the GWAS. It's, like I said, I have done many myself and just the statistical sample size, and I will not be technical in any way. But the experience is that the more you look, the more you will find. Mm-hmm. And so what you have is this army of genetic variants. I know you term them as genes, but the effects are so small. Mm-hmm. So where does one begin? Because in aggregate, maybe they explain maybe 10% or...
Absolutely. If I may, I have a slide that shows exactly what Dr. DeVivo is saying, that you keep identifying more and more genetic variants that influence these disorders as the sample sizes increase. But what we found is that we're actually already reaching the plateau of the number of cell types that are associated. So whereas we see continued growth in the genetic variant discovery phase, it seems like we may have already found the majority of cell types that are relevant.
So would you say that the most surprising result was this idea that the genetics were similar across males? Well, what surprised you? I don't want to put any in.
Sure.
What surprised you about your results?
Right. I think the thing that has been most surprising to me is that this approach seems to work so well, despite the fact that the model is so simple. So there's a lot that we can do to improve it, but when we look at more and more complex brain conditions, we keep finding what seem to be true positives. So I think what's been surprising is that we didn't think that something so simple could work this well.
What do you think is missing from this analysis that you could add?
Actually about half the genome is missing from this analysis.
Yeah.
So to be seen what we find when we add that back in.
Okay, so we need part 2 and part 3, and
yes.
But I want to leave time so we can have the full discussion that I keep threatening you all with. And so with that, I'd like to bring these fabulous speakers, Tara, Victoria, Étienne. So there are many, many questions that have come through the Slido, and I use this opportunity to remind everyone sitting here in the audience as well as at home to send questions through the Slido contraption here. Okay. So I wanted to ask, I felt I was a bit short with Tara in some of the questions that I wanted to ask. And I want to start with you before I go to the questions. So what I really wanted to talk about, and I briefly mentioned it during the break, but I bring it up now. When I saw the trends that still women are being left out, and even though it's getting better, it reminded me of a very great story that I'd like to share, which is related to those numbers as well as the hysterectomy question that I'm going to ask you, which is when I came to Harvard from Stanford, and I was always a cancer person, a researcher, and I was going through the databases and which cancer am I going to study in the nurses' health study. And I really was intrigued by endometrial cancer or uterine cancer because it had these exquisite risk factors. And as someone who works in the lab and trying to understand etiology, sort of like these are clues as to where you should be looking. So I remember running into a senior faculty member, and I said, I think I know where what I want to write my first R01 on. And I said, I want to study the genetics or the genetic susceptibility to endometrial cancer, uterine cancer. And he looked at me and he said, very quizzically, and he said, a smart girl like you, why would you want to waste your time on a cancer that any woman can get their uterus taken out? And it's done. And I said, oh, okay, well, I'm going to do it anyway. And that's what I've done. And so my question to you, Tara, is I would assume now, because it was a trend where women were being told in their 40s to get their uteruses taken out, how does that impact the hysterectomy rates in your catchment area?
That's a great question. I mean, obviously, I think that experience you had just reflects great misogyny and the over tendency to generalize women's health and the complexity of it. And for a long time, there was a lot of paternalism in medicine. Most OBGYNs were male, and if you had any complaint, just take it out. You know, who cares if you went in menopause if you were 30 or 40? I think, so historically, hysterectomy rates were pretty large. It was a pretty blunt tool to address a lot of conditions in women. And over time, we've become more thoughtful. There's more women that are OBGYNs. The practice of paternalism isn't gone, but it's been addressed somewhat in some spaces better. So we do see hysterectomy rates going down. I think there's some paradoxical findings because we now find the endometrial cancer rates going up. So no, I don't recommend using a chainsaw and everyone getting a hysterectomy, you know, at perimenopause or menopause. But I do think it does show that maybe we were masking some biology. And then over time, you know, we know that endometrial cancer, not all, because there's endometrial cancers like 20 different cancers, but the most common type of endometrial cancers are very environmentally driven. So obesity, diet, lifestyle can really be drivers of those cancers. But certainly there's other types of endometrial cancers that occur and based in biology. So I think that we were masking a lot of it, but also we were masking women's symptoms. And I think now we still need to do better. I see women after they've become diagnosed with endometrial cancer, and sometimes I worry the pendulum swung too far. You know, we're really hesitant to do surgery. It's so invasive, it's so aggressive. And I have so many women, and specifically Black women, that say, I was begging for a hysterectomy, and my doctors kept saying, oh, you don't need that, you don't need that. But I'm still bleeding, and I'm 55, and I'm, you know, oh, it's fine. And not just understanding the biology of women, you know, I think we have to really listen to women, and I think we need to know what's going on. And I think menstruation and all those things that are thought to be impolite to talk about in certain groups is really important. It's an important vital sign for your health as a woman. So, you know, when we unmask that and we leave everyone's uterus in, we do see an increase. We have an increase. It's one of the cancers that we're seeing an increase in. That might be due to obesity, but there's some other things that we can't just chalk up to less hysterectomies and more obesity. There's some other interesting things going on.
Great. That was a very insightful response. I'm going to just ask one other I mean, there are many questions, but this question has been asked multiple times. What is a virtual nursing program, Tara?
Yeah, thank you for asking. So one of the fallouts from the COVID pandemic was that there was a huge workforce shortage. And I think a lot of clinical trial offices across the country were kind of having just a flattening of their ability to open new trials, to handle the regulatory load. And this was kind of a response to even have clinical trial research coordinators and navigators, no offense, but they were being poached to pharma jobs where they had better quality of life, better pay, they could work from home. So people weren't actually, it wasn't very appealing to come work in academics and be a clinical research nurse or a clinical research coordinator. So we had to do more with less. And one of the ways that we did that was say, well, some of these trials, it is not explicitly stated that the patient and the navigator or coordinator have to be in the same room signing the consent with wet ink. You can do all the counseling remotely. Patients can get all that done. So we said, okay, we have 50 sites. We don't have 50 clinical research coordinators, but if we assign a few to be available via, audio-visual app to talk to patients when the busy clinicians are running from room to room, making diagnoses, making plans, those clinical research navigators for the protocols in which it was eligible were able to do that and effectively consent patients, and then they'll get back with the investigator or the provider and say, okay, she consented, and this is the next steps for clinical trial enrollment. So we've been able to do that. I have a few sites where I work with my virtual nurse navigator.
Okay, well, great. I'm going to move to Victoria. Again, I had some questions that I reserved for myself, and then I will go to the Slido. And it relates to the market and not wanting to support precision medicine just because, resources are tight, so maybe we should focus on allergies and inflammation. So the question I have is, are there successful examples or emerging breakthroughs that could shift investor sentiment in favor of precision medicine for ovarian cancer or any other cancer?
Yeah, that's a good question. Yes, I think that if there are a few major wins here, it may be able to shift investor attraction back to precision medicine. Very recently, a smaller biotech, Scorpion Therapeutics, was just bought by Eli Lilly for almost $2 billion for one asset, which was a precision medicine breast cancer drug. And to be fair, that patient population is quite large. It's a lot bigger than cyclin amplified ovarian cancer. But that's an example of a successful, or hopefully to be successful, precision medicine therapy. So I think if we have a few more of those, kind of back to back, maybe the winds will shift.
There's another question here. How can precision medicine address the rates of ovarian cancer among women of color, considering the historical injustices and disparities in healthcare access?
Yes, also a good question. To understand a patient's genomic alteration, they need to have access to testing. And I think we've learned from all of the talks here today that can be challenging for some individuals in certain communities. And so a lot of the data that we have is probably skewed toward women who live near big cancer centers, for example, or women who seek medical care frequently, or women who maybe just have more knowledge of what's going on, just from their educational background. And so there needs to be some systemic changes there. Drug companies do have initiatives to reach women who are in disadvantaged situations, but that's obviously not going to be enough. That needs to come from either like grassroots organizations or some kind of government intervention, but I'm not sure if right now we're going to be able to see those types of changes.
And that, Étienne, I had a question for you about, you brought up the redlining just briefly in your talk, and would that be your next step? In the research, it seemed to me that's the direction you're going to go into.
Yeah, one of my next steps for my research is to look at historical redlining and how it impacts breast cancer risk as well as survival. But I'm also really interested in also delving a step further and thinking about how where we live and our experiences, how that gets embodied and how that impacts like the epigenome, for example, and the tumor microenvironments. So those are some of the next steps that I hope to delve into.
That's a really good setup for me because here's the next question, which sort of comes close to what you're going to be doing next. So given the correlation between experience of racism and negative health outcomes, what is the possible causal pathway?
Yeah, that's a great question. And, as epidemiologists and all of us researchers in this field, we definitely think about the causal pathway and where we can intervene. I think there's some different pathways, definitely inflammatory pathways, where this can be impacted. It can, impact the immune system, these psychosocial stressors, and just your overall quality of life and health.
So another question for you. Do you have information on healthcare spending, RE outcomes for populations studied? How much does spending on care from any other source matter?
The Black Women's Health Study, I don't believe we have information on healthcare spending, but I'm sure that could potentially be garnered through some sort of linkage to an agency that has that information. So that maybe we can see in the regions in which our participants live the amount of healthcare spending on specific diseases or outcomes. And I think that potentially directly is aligned with the resources that area has in terms of what they can offer different patients potentially in terms of healthcare spending. But that would be an interesting area to delve further into.
Do they ask that on the questionnaires? I don't think so.
No.
They don't ask.
Oh, individual healthcare spending?
Maybe.
Okay, maybe that's where they, no, they don't ask questions on how much the participants spend on their own healthcare.
Yeah, so you have to go, like you said, regional or...
Yeah, more regional estimates. I mean, they do ask about, health insurance, so we know what type of health insurance or whether they're insured or not, which is an important, proxy in terms of how you're maybe utilizing the healthcare system or your ability to utilize the healthcare system, but not individual spending on healthcare particularly.
Yes. So going back to Laramie here, I'm going to get my questions in. Drug targets. So my question was related to, do current medications for schizophrenia target these neurons that you showed?
Yes, so as far as we can tell, they do target just a couple of the neurons that were associated with schizophrenia, but not most of them. So it looks like maybe they're modulating some of the circuits that are important for schizophrenia, but probably not others.
Are you working with companies or departments where they can make these compounds based on the findings?
We're just starting that work, and I'm just learning how to go about that.
Okay, so here's a question for you, Laramie. Is there a risk to the integrity of long-term studies? Oh, wait, that says Laramie, but I think it's for Black Women's Health Studies, so I'll come back to you. Laramie, is your GWAS international or is it internal to US samples only?
Right, so this GWAS is international, but it's not representative of most populations. So unfortunately, the only GWAS that was well enough powered for this analysis was exclusively in European ancestry populations. So a lot of my other work has been focused on this now well-known problem of the GWAS studies not being representative.
Right.
So it's representative only of sort of wealthy white populations.
So I want to follow up on that. So did you take already studies in hand that were available? So unfortunately, a lot of the studies that are available were based on participants who are of European ancestry. So going forward, there is a large effort, there was a large effort to start cohorts and populations that would include, yes. So there's a question here again, Laramie. I would love for Laramie to say more about the study approach process that is discerning different cell types.
Sure. So it's a two-step process. One part is a biological assay, and the other part is computational. So the biological assay, the important thing that happens is that cells are separated from one another, And then the technology is able now to look inside those cells and see what genes are expressed and at what levels. So that's the biological assay. And it creates basically information for each individual cell. And then there are computational approaches. This is step 2. These computational approaches are applied to the data. They're applied to the information that we have about what genes each cell is using. And when you apply lots of different computational techniques, they all tend to yield similar clusters of cells that go together in broader categories and then more and more fine-grained categories. So that's how these cell types are determined. There are certainly a lot of details along the way, but that's the general approach.
I want to be very fair, so I'm going to just use the same method of asking the question in that order. So I'm going to go back to you, Tara. Doctors on podcast want to engage women's health from the prevention angle. How do we work to help industry, established systems help truly partner to educate?
I mean, that's great. There's a huge appetite from the public, right, for women's health information. If I go on TikTok or Instagram and I search hashtag IUD, hashtag menopause, you're going to find a wide range of sources. Some of them will be physicians, some of them will be epidemiologists, researchers, but a lot of them will just be lay patients with their experiences, influencers. So I'm a little bit averse to going out and, my goal is not to be like Instagram famous, but I think a part of your job, if you're in this sphere, is to try your best to put yourself in those spaces, just to be another voice of evidence, of experience, and to listen. I think that's a challenge. I think I train a lot of younger residents that are going to be OBGYNs in the community, and this is a lot of what we deal with, when patients come in and talk to us. So we have to be present. We have to know what patients are getting exposed to outside of the hospitals and the clinics and the information and oftentimes disinformation. So I think it's part of our job now to be aware of that side of it, especially when we're caring for women and other vulnerable populations, for sure.
Yeah, I follow up my own. How do you train and model your trainees? I mean,
don't ask them. No, I try to model. I try to just really stop and say why I do what I do. This is why I'll take. I had lots of people model this to me and how to listen to patients, but really it comes down to listening to patients, taking time to educate patients. And I just try to say out loud what I do. And I try to actually just demystify research, I think, for a lot of them, because it's a hard career already, and then asking someone to go into research. And I think sometimes in research we can gatekeep a little bit. It's very complex, and it's a hard thing to break into, especially if you're coming from the clinician side. So I try to just model that this is exciting. I do it because I don't do it for the money. I don't do it for the, the prestige, but I do it because it makes an impact. So I really try to say, this is cool. I'm putting this patient on this clinical trial, and look, this clinical trial led to this approval and this drug that's now saving lives for endometrial cancer patients. So I take a little bit and say, there's the little bits that we can do in our life, whether it's research or patient education or advocacy, try to model that forward. But I'm not perfect. We have bad days, and we just try to, always be open and honest.
One other question, because it's a recurring question actually for many of the speakers, which is, not surprisingly, what is the state of the moment for our scientific research community in relation to federal funding cuts, grant freezes? I'd like to hear it directly. That's not my question, but a very poignant question that came through.
I think I'm the only person here from a red state. So I think I haven't gotten the overwhelming sense that the actual substance is what's being questioned. A lot of this is just political performance, and the value of our work, the people around you say it's still important. We still want to support it, whether we have to not use certain words in certain contexts, and we have had funding pulled. Funding gets pulled, and it might come back. I think that's a real unsettling thing, just to dismantle federal funding and the support and the workforce that it supports, even down to your, hospital systems and the researchers that feed into these hospital systems. It's going to make an impact, and it's been hard. It's been disheartening because nothing moves fast in research, and when you take things away, resources away, without being thoughtful, I feel like we're gonna set ourselves back we're always working on progress, and once that progress, hiring freezes and things like that, it's disheartening because you feel like the progress that you are working, the gears are gonna stop, and we're gonna have to rebuild all this. So I think as an individual and a system level, it feels a little bit like dread, that a lot of the work we're doing, we're gonna have to pause and come back to it. And things are moving so fast in science around us, and they're gonna keep moving in other countries. They're not waiting for us.
Right.
So I think that's the disheartening part.
Victoria, I have a very specific question for you. Do the PARB2 or BRCA inhibitors that are already known to work in breast cancer patients as effective in ovarian cancer treatments?
I don't know if I can speak to the differences in efficacy in breast and ovarian cancer, but they have been transformative in breast cancer for sure. And PARP inhibitors are in use in ovarian cancer. But yeah, I don't feel equipped to answer.
I can answer that perhaps because I prescribe PARP inhibitors.
This is a group discussion. Please chime
Yeah.
in, everyone.
PARP inhibitors have been transformative for ovarian cancer, and part of my job at diagnosing ovarian cancer for a patient is to really give them that hope that we have all these new drugs coming out, these targeted drugs, and there have been quite a bit of wins even in GYN cancers beyond PARP inhibitors. But I tell women this cancer is now considered a chronic disease. You will be alive in 5 years because of all these things we do, and hopefully you'll be cured. And PARP inhibitors are really, we're careful to say it, but it's really the other C word in cancer is cure, and we're curing women with these drugs. So it's really exciting. But like Victoria alluded to, it's only a minority of all of our cancer patients because there's other drivers and molecular drivers for ovarian cancer.
Victoria, are you can you explain the different phases of the clinical trials?
Sure. So in oncology, a phase 1 trial, the aim of that trial is to establish safety of your drug, not so much efficacy. So the idea is that you enroll patients in different buckets of increasing amounts of drug, and then the idea is to reach a max tolerated dose. And then from there, you can decide your recommended phase 2 dose. And the phase 2 trial is more looking at efficacy. And then phase 3 will build off of that in larger sizes. And then hopefully after that is approval. But I will say at any point in that process, the failure rate is extremely high, especially if you're comparing your drug to standard of care. Oftentimes there's not a meaningful difference, and then there's not really a point in moving forward with your targeted therapy. But yeah, that's kind of a really fast overview of the different phases and the aims of each phase.
And it doesn't always, this is part of the reason why it costs so much money and time and takes 20 years or 10 years, because there's no guarantees, even if you go through phase 1 and phase 2.
Yes, the failure rate is so high. It's like over 90%. It's, you want to fail early in drug discovery. So ideally, you'd like to fail during target validation stage, like early in the lab, before this drug even has a thought of entering a patient. But oftentimes, companies move drugs forward into the clinic, and some unforeseen things happen. And yeah, they just don't make it.
Étienne, there's a question here for you. Considering the time we are in, how have you found success talking about women's health in perhaps new or different ways?
What was the last part?
Considering the time we are in, how have you found success talking about women's health in perhaps new or different ways?
Well, I'm really passionate about this work, so I just enjoy engaging people and talking to people about this work. I, have done it at conferences, but just in casual conversations with friends. And this is just even, friends who maybe have different ideologies or different, irregardless of that, I still, like to share with different individuals. I think also spreading it on, I don't do a whole lot of posting on social media, but I do think it's important to share the studies that I'm publishing or the new findings to the public via social media. Not that I have a following, and I'm also not trying to be Insta famous, but I like to be able to be the one to communicate my findings to a broader community so that there's no misinterpretation of my study per se or the work that I'm doing. So I think that could be a new and exciting way as well, but I think I haven't thought too much in that area, but there's definitely work that can be done because I think it's important to get this message out broadly.
All right, some of the grassroots
Yeah,
efforts
some
that
of
you
the
were mentioning,
grassroots
going into the
efforts.
community.
Right.
I mean, some of the work I know the Black Women's Health Study has done is going into churches and community centers and really to get to where the community lives or socializes.
Yeah, so I haven't done some of those presentations myself, but yes, like you said, there are some of the PIs and research staff will go into the communities, different churches, give presentations, talk to different groups, which I think is very important.
I have another question regarding mobility and health outcomes in southern states. Southern states tend to be totally dependent on highways. Is there a movement to shift away from highways?
I don't know if there's necessarily a transportation movement to shift away from highways. That's again one of those larger public health infrastructure interventions that you need to slowly chip away at. Perhaps it would be nice if cities were more interconnected, but I think in the meantime, while we're trying to reconstruct some of these communities, perhaps starting with better transportation to connect individuals to different parts of the city would be helpful, or just bringing the resources to them. Let's make clinics in some of these areas so we can actually bring the resources to where the people are. But I don't know specifically about the transportation.
Yeah, historically, the highways were put in areas that were, as you noted on the map, not desirable, right? I mean, so it all makes sense that these would also be the areas where you would need the most intervention. And so, Laramie, you're up next. The approach for schizophrenia, can it be used for bipolar, which can sometimes resemble schizophrenia?
Yeah, great question. Yes, it's exactly the same approach. And we did apply it to bipolar disorder. And kind of as this question implies, we might expect that we would find some of the same cell types and a few different ones, and that's what we see. And we're just starting to kind of delve into those results.
Are the cell types found in psychiatric disorders correlated to environment or diet?
Right, so with this approach, intentionally we used just a healthy human brain sample for definition of the cell types. So we're looking at just what is the set of cell types in a quote-unquote healthy brain. So they may be influenced by diet, or they certainly are in different ways. But what we're testing is just, we're assuming that these cell types are functioning properly, and we're asking what genes they use.
Here's another great question. For psychiatric conditions, given difference in occurrence rates but similar DNA behavior, is it possible that fractions of cell types differ by gender?
Yes, absolutely. There's actually a researcher here in Boston who does beautiful work looking at rodents and using these same approaches to identify the cell types in rodent brains. And she found that there's a neuron type that doesn't exist and isn't detectable in male mice until they have pups. Until they're a father, they can't detect this neuron type, but they find it once the male rat has that experience.
So it's like developmental, some trigger or...
Developmental and some trigger Yes.
Well, that's
So I'm
not-
sure there must be some differences. It's probably mostly the same, but then there will be important differences.
Excellent. I have to go back to you, Tara. There is a question about post-hysterectomies, and I'm going to look for it because this is a personal concern. Post-hysterectomy, repetitive occurrence. Oh, post-hysterectomy, repetitive recurrence of endometriosis? I don't.
Yeah, it can happen. So I think maybe the question is like after the uterus is removed, you can still have endometriosis. So I think endometriosis is a very perplexing condition. It's understudied and underfunded, but it has a big impact on a lot of women. And even though it it would make sense, right, to the layperson, no, you remove the source of the endometrial tissue that's implanting into the belly, into the pelvic organs, then you remove the source, then the endometriosis will stop. Well, we know that that's not true. You can do a hysterectomy and women will still suffer from side effects of endometriosis because it's ectopic endometrial tissue on the peritoneal surfaces, on the ovaries, so really, unfortunately, it's very hormonally driven. So if you still have the ovaries in situ that are still, cycling through estrogen and progesterone, then you'll still get a cyclical response and have the endometriosis symptoms. So, I mean, this is a real important thing. I think just talking to a patient about it, talking to a colleague or a researcher, it's complicated. And I think women's health is understood. I don't think I worded that well, but it's poorly understood, and it's just simple things like that. Even explaining women's health to a woman can sometimes be so complicated, and women are like, well, why am I still having this issue if this was removed? And doctors are terrible. I have so many women that don't know what female organs they still have left, and they've gone through surgeries. It's confusing and complicated, so I think we need to just talk about this more, draw pictures,
That reminds me, and I know we're almost short on time, another really interesting anecdotal story where I was doing an outreach at Sloan Kettering with endometrial survivals, uterine survival cancer survivors, and they were African American. And what was stunning to me was many of them, the three of them that were there, that's why I'm saying many, because all of them had the same story, which was when they had symptoms for endometrial cancer, and one of the main symptoms, as we know, the bleeding, they were ashamed to tell their clinicians or their nurses or whoever their healthcare provider was, because they confused endometrial abnormalities with cervical cancer. And so there's this, I'm going to go colloquial, crazy idea that cervical cancer, there's some sort of judgment or shame. And this is what they said. They felt ashamed, or they were going to be judged if they went to the doctor to ask about these symptoms. And because they thought they brought on their own cervical cancer. Now, I don't want to go into the disease itself, but that's sort of like, it triggered that story in my mind, which is what you said, that women themselves are not empowered to even ask these questions because of societal, you can speak better to that, but.
I mean, I agree. I think it's very uncomfortable sometimes to talk about things we don't talk about commonly. Yeah, I have a lot of patients that, get history and they're not quite sure if it was endometrial or ovarian or cervical cancer that their family member had because they just didn't talk about it. But something happened to one of their aunts or their grandma that had, required a surgery. So I have to kind of tease it out, like what kind of surgery, what kind of chemo, did they get radiation? I got to try to figure out exactly the most likely cancer. I think it goes across racial and ethnic barriers. I think there's some socioeconomic stratification, that goes with that. But yeah, I think there's some shame attached to it for so long, like the HPV vaccine, right? There's not huge uptake because, this is a vaccine that can prevent cancer. We know that's the truth, and it does, and we have the evidence that the incidence of cervical cancer has decreased. But people don't want to get the vaccine, not just because the hesitancy to trust in vaccines, but because there's some implication that it's associated with sexual activity, So there's just a lot of traps you can walk into when you're talking about these things, and you just have to ask, why do you think this? What is your concern? And it's hard. It's hard to navigate for us as clinicians who talk about this every day. So I can imagine for a lay patient who maybe had a few bad experiences in the medical field, with interactions, it can be really daunting and challenging.
Thank you. We have time for one more question, and I'm going to end it with Victoria. The role of macroeconomic factors and how that influences investor decisions for funding precision medicine writ large or focused on women of color in particular.
So is the question, how is that affecting?
What role do macroeconomic factors play?
Yeah, so I think, again, right now in drug discovery, the investor decisions are going to be on sure bets with large patient populations. And so if there are like niche indications that primarily affect women or women of color, those are going to be underfunded because they are nothing compared to huge patient populations for these other types of indications like weight loss. And so it's going to go, it's just the way of economics, I guess. So we're going to need an economic turnaround, I think, to re-excite investors and to re-excite companies to put money there to make an impact.
So it's the market. That's
It's the market, yeah, always.
Yeah. Well, I could go on all day, but I know people have places to go and people to see. So I want to thank this brilliant panel of speakers, Laramie, Tara, Victoria, Anthony, and I want to thank you all in the audience and everyone at home. I want to thank Ito, and I want to thank Becky and Don and the entire Radcliffe staff, and our dean, who's always so supportive, and a special thanks to Melanie and David Nemec for making today's event possible. Thank you, and have a good rest of your afternoon.